Hey everyone! In today's video, we're going to be looking at a common mechanism in which messenger molecules get released. So, in the past few videos, we've been focusing on this cell signaling framework, where first messenger molecules are produced, then they are sometimes packaged, and then they get released, produce a response, and the response must be terminated. In the last video, we focused all about termination, but today, we're going to be focusing all about release. After a messenger molecule is packaged, how does it get released from a cell? Well, this is due to some important proteins known as VAMPs/V snares or SNAPs/T snares. So let's take a look at how these proteins work together to allow for the release of messenger molecules. Let's get into it. Okay, so messenger molecules can be stored in a few different types of cells. One type of cell is called the mast cell. So mast cells are cells found in connective tissues all throughout the body. And mast cells, they're involved in immune responses and inflammation. They're basically storage cells. They store messenger molecules such as histamine and other messenger molecules that are related to immune responses and inflammation. So things such as injuries, different chemicals, or antigens. Antigens are things that trigger immune responses. So in different injuries, chemicals, and antigens, they can stimulate the release of messenger molecules that are inside mast cells. So basically, let's say this is your mast cell, right? Inside your mast cell, you have these little storage compartments called granules. And inside these granules is where you have your stored messenger molecules. So for example, histamine. So I'll draw little H's for histamine. And then when things that stimulate the release of these messenger molecules, such as let's say an injury or chemicals in the that enter the body or different antigens, when these things can stimulate the release of these messenger molecules like histamine out of the mast cell. And then after they're released, they can go and do their responses. So another type of cell that can store messenger molecules is the neurons, which are cells that act as a part of your nervous system. So neurons, they can communicate with other neuron cells or other types of cells like muscles or glands via something called the synapse. So basically, right, you have a neuron like that, and then you have a gap called the synapse. And then after the synapse, you can have, let's say, another neuron. So what can happen is these two, the neuron that comes before the synapse is called the presynaptic neuron. And the one that comes after the synapse is called the postsynaptic neuron. So your presynaptic neuron can contain neurotransmitters. What neurotransmitters are they're basically messenger molecules, but messenger molecules that are specific to neurons. And these messenger molecules can be stored in little compartments known as vesicles. So remember before with mast cells, your messenger molecules were stored in little components called granules, whereas with neurons, your messenger molecules are stored in vesicles. So very similar, it's just, think of it just a change in name. With mast cells, you call them granules. With neurons, you call them vesicles. But your neurotransmitters that are stored in vesicles, they can get released and go out into the synapse. And when they do, they can act on your postsynaptic neuron. Or let's say instead of a postsynaptic neuron, let's say you have a muscle or gland. It can act on those. It can go and bind to receptors and produce a response. But today we're just focusing on release. So let's take a look at how mast cells can release messenger molecules from granules and how neurons can release neurotransmitters from their vesicles. One quick thing to note is that it doesn't always have to go from your presynaptic neuron to your postsynaptic neuron. In fact, sometimes the signaling can go backwards. So your postsynaptic neuron can release a messenger molecule out into the synapse, and then this can go and act on your presynaptic neuron. So the signaling can actually go backwards. And this is what's called retrograde signaling. It seems weird, but it's possible. But either if it goes from presynaptic to postsynaptic, or vice versa, the mechanism of release is pretty similar. Okay, so here you have your mast cell with a messenger molecule inside a granule. And here you have your neuron with a neurotransmitter inside your vesicle. So the good news is that the release mechanisms of both of these are pretty similar. So basically you have different proteins that are present on the membrane of the cell and on the membrane of the granule or the vesicle. So attached to the granule or the vesicle, you have proteins which are called vamps / V snares. These proteins, for now, they may have some slight differences, but think of them as being pretty much the same. Same here, you have vamp / V snare. And then on the cell membrane, you have different types of proteins. And these proteins are called snap proteins or T snares. Snap or T snare. And an easy way to remember which is which, you can remember for a vamp / V snare, they both have a V. And V, you can remember vesicle. So vamps and V snares are found attached to the membrane of your vesicle, whereas the other one, snap / T snare, is attached to the membrane of your cell itself. So these proteins basically interact with each other. So your vamp proteins, they interact with your snap proteins in order to release your messenger molecule from its granule or release your neurotransmitter from its vesicle. So let's take a look at how they interact to allow for this release process. So your release process is generally made of the 4 following steps. So firstly, translocation. So your granule or your vesicle needs to move and make its way closer to the cell membrane to allow for release. Then comes tethering. So your vamp or V snare proteins, they link up together with your snap and T snare proteins. Then comes docking and priming. The vamp and snap proteins continue to interact with each other, and it prepares for the final step of exocytosis. And then comes exocytosis or fusion. Basically what happens is your granule or your vesicle, it fuses together with your outer cell membrane, and this fusion releases your messenger molecule or your neurotransmitter out of the cell into either the extracellular space or into the synapse. So let's go through each of these one by one with a diagram to make it a bit more clear. Okay, so the first step, translocation. As you can see, your granule containing your messenger molecule is moving closer to your cell membrane. And this allows for the next step of tethering. So as you can see, your snap and vamp proteins are interlinking together. And then in the next part of docking and priming, there's a lot more interactions between your snap and vamp proteins and your your granule, which contains messenger molecules, is getting even closer to your cell membrane as it prepares for the final step. And that final step is fusion or exocytosis. So your granule membrane fuses together with your cell membrane. And in doing so, exocytosis happens. Exocytosis is when something in the cell is released into the outside, and that's what happens with your messenger molecule. It gets exocytosed and leaves the cell, goes into the extracellular space where it can go and do responses. Now, if the part about the fusion of the membranes and the release of your messenger molecule is a bit confusing, I'll show a diagram here. Hopefully this makes it a bit more clear. But generally, this is your pathway: translocation, tethering, docking / priming, and fusion and exocytosis. And this is applicable to both granules inside mast cells as well as vesicles inside neurons. So one more thing to note, specifically with neurons and neurotransmitters, in order for this final step of fusion and exocytosis, where the membranes fuse together and the neurotransmitter is released out into the synapse, you need the influx of calcium ions. So as you can see here, the SNAP and the VAMP proteins are all linked up together. This is the docking and the priming stage. So we're like just about ready for exocytosis and release, but it relies on the influx of calcium ions through these ion channels. Only then can fusion and exocytosis happen. So that brings us basically to the end of the video. I hope this made the release mechanisms of messenger molecules from mast cells and neurons a bit more clear. So I hope you were able to understand these different 4 steps of translocation, tethering, docking, priming, and fusion exocytosis, and also understand that with neurons, sometimes it's a bit more unpredictable, and messenger molecules can travel backwards from the postsynaptic cell to the presynaptic cell via retrograde signaling. And also understand that with neurons, the release, or especially this final step of fusion exocytosis, is reliant on the inflow of calcium ions. But that's about it. Release is one of the more simpler steps. But I hope you guys enjoyed the video. Thank you guys so much for watching. If you enjoyed or you found it useful, please consider leaving a like or subscribing. I'd really appreciate the supports. And if there's anything you would wish for me to go over again, or any tips or feedbacks or other comments, please feel free to leave a comment down below. I'm open to any suggestions and I'd really appreciate it. But until then, I'll see you guys in the next video. Take care for now. See you.